Well, thank you everybody for joining us today. We've had an ongoing effort here over the last, well, it's been over a year really, to really replace our SeqMan Pro software with SeqMan Ultra. SeqMan Pro, has been around since, really for almost 30 years. So we've had a lot of users and we've gotten a lot of feedback from them when we release SeqMan Ultra on things that they would like us to bring back from SeqMan Pro, some behaviors, some new capability. So it's really been an ongoing effort back and forth with our customers to really improve this software, and it's continuing now. I'm actually going to be showing you a version that's we're going to have another update out late at the end of this year. So it's a kind of a continuous process, and it really is important for us to get the feedback from our users, to keep making these improvements. So we're going to be looking at SeqMan Pro, which is part of the DNASTAR laser gene suite, which does both molecular biology. So we consider some of the small Sanger projects molecular biology, and of course it's also used for genomics. And we've really kind of merged the two in SeqMan Pro, so it's a little bit more seamless than it was, or excuse me, in SeqMan Ultra than it was in the past. So in particular, some of the new features that we'll be looking at is a quick way to assemble Sanger data, to preview it, trim it, and then run an assembly. So this is some of the feedback that we've gotten where it was easier in SeqMan Pro. You could just dump some reads in and with a couple of clicks have your assembly. And so we brought that kind of fast Sanger assembly back. One of the issues with that approach, though, is that in some cases, the scale or scope of the project was larger than what SeqMan Pro could handle very well. For example, you might align a few Sanger reads to a human chromosome. That really requires a different high-capacity type assembler and a little more input from the user. So we've made it such that we can detect when the project is much larger and then redirect the user. So that's part of this integrated wizard. So for larger NGS workflows, we've integrated our SeqMan Engine Wizard into SeqMan Ultra, so we can guide people down, through that wizard. We've also introduced a jobs panel management. So when you're running longer running jobs, so this would be really any NGS or larger assembly, or something like a blast search, there's a jobs panel then, or a cloud assembly that would allow you to monitor the progress of those jobs and work in the program while those jobs are running. So it's a nicer way to keep track of multiple things. There's also a search bar that we've brought in right on the toolbar. I'll show you that. Then there's also an option that was really buried in SeqMan Pro, but it's very, very useful for some workflows, and that's the option to assemble to parse similar sequences. So rather than using the assembler to try to lump everything together in a config, to use the assembler to try to separate similar sequences into their own config. So it's a different way to use the assembler that was there in SeqMan Pro, just buried in the advanced parameters. We brought that right out in the surface for those workflows. And we've also introduced consensus and reference ruler tracks. And it seems very basic, but it's essentially when there's a reference, there's a coordinate space for the reference. And when you align reads to it, that introduce gaps or delete or insertions, it's a different coordinate system that's called the gap coordinate system or the consensus. SeqMan Pro didn't really delineate with that very well. So, one of our most common tech supports over the years was people were confused by which coordinate space they were in. So we just put two ruler tracks, kind of like a browser, and you can clearly see when you're in reference space or consensus space. So we'll take a look at that as well. And then, of course, exporting our images out. In our other applications that we call our newest applications that are in this RCP rich client platform, we have the ability to export in bitmaps, PDFs, or even in PowerPoint format. And we make it much easier. So these are kind of the new things that we're working on that we have in 17.2. We have some new things, again, a few more updates that you'll see later this year, and we're just things are just moving along here. So I'm gonna open up the software and just kind of show you some of these new features. Now there is some with the webinar running. There's a little risk here. I've got a development build essentially, so please bear with me if we have any technical snags. I think I can try to avoid all of them, but this is still hot off the press software. So again, with SeqMan Ultra, we have a welcome screen, and you can see that under new assembly, we have a couple of options here, new Sanger ABI, and a tooltip just shows up that I can click on this button or drop files there. So if you're used to drag and drop, you can drag and drop ABI reads right onto this button. It'll load them up and you'll be ready to go. There's also a new assembly with options, and this is for more complicated type alignments of Sanger data. If you've got multi-sample and you want to assemble by name, or if you're going to align to a chromosome sequence, something much larger, you'll go through a few more pages in the wizard to set that up properly. So this entry point looks different. You can also see that for our NGS customers, we have the NGS workflows here. We still have the open assembly, so it keeps track of what you've been working on lately, and you can open those. So I use these two tabs all the time now that I'm used to the new software, and it really saves a lot of time searching, opening existing projects and launching new workflows. This to show you the difference here. I'll click on new assembly with options, and you can see it opens a window now, and this window is really an assembly wizard, right? And so I can set up any type of an assembly that's more complicated, using this approach. Just like if I select one of the genomics workflows, if I select, de novo genome and editing, it'll have the de novo genome assembly and finishing workflows. And again, this would allow me to do Sanger, NGS, or long read type workflows. But now what's new, so that wizard then is integrated with SeqMan Ultra. And if I start those jobs, if I start a big assembly that's going to take an hour, there's this jobs panel in the bottom right here, and it'll show up in that jobs panel, and I can monitor progress, and when it's done, there'll be a link to open that. So I can do multiple things. Now, if I'm just doing Sanger, I can click on that option, and you can see a new window appears in SeqMan Ultra. And this window then is modeled after the unassembled window that we had in SeqMan Pro. And this is meant to give us all the tools that we need to assemble Sanger trace data. So I'm going to use this add button to add some sequences. So I'll just grab some ABI files here. I'm going to move this little webinar controller out of my way. Okay, so I've loaded in 14 sequences in this case. You can see there's down at the bottom here, I'll expand this. There's some little hint text, select on table to open trace, single click in trace to trim manually. So as I make a selection, now we have a previewer of the trace data. So we had gotten pretty strong feedback that folks still like to look at the trace data before they do any assembly. And some people might just manually want to trim. So I can look at the traces. I can add quality scores. I can make these, so I'm just making this bigger here so we can see these traces. I can scroll now. There's also some trim bars here on the edge, so I could grab them and drag them and put a manual trim on my sequences on both ends. Right, and so for some projects, some folks just prefer to do it that way. You'll see that as I trim up in the table, those coordinates then are placed in the limits. I've set limits on the sequence now. And then I can reset it and look at another trace. And there's some other trim options here. There's quality trim options. So again, our trimmer will look at windows and get a quality score. So I can separately do a trim. So I'm just going to use this trim button to trim now, and it'll do a quality trim on all the reads. You'll notice that as I click on them, this single click, it updates, and some have quality trim at the front and some do not. Now I know that this particular data has came from a vector, and so there's also a vector trim option. And if I just go ahead and trim now, you can see we get this orange zone now, and I'm going to zoom out a little bit. So the orange highlighted is areas where there was a vector detected and where it trimmed, right? So I can see then the difference between the quality trim, the manual trim, and the vector trimming. And it tells me which vector. So what happened there is I didn't set a vector. It's just using our set of vector sequences that we have in the data location. So that's the default. It's just, and you can see that some vectors that are really close, this inverse Janus and this phi X 174 are very, very similar. So it used two different vectors to trim these sequences, right? But if you know what the vector is, again, I'm going to reset the trim here. I can also set the vector to a specific sequence. And I know that these have a vector called Janus in them. And so now if I can set the vector, trim, and now I have my vector trimmed. I can see the coordinates of the trim. We use this little nomenclature with brackets that show, trimming, what the vector trimming did, what the quality trimming did, and then it is ready to assemble. So lots of ability here to preview the sequences, trim them, and then assemble. You can also see that the match percentage, which is the single most important parameter for assembling, is now exposed right on the toolbar. And it's set by default at 80% for Sanger data. And if you're going to change one parameter, that's the one that I would recommend. You can make it more stringent by increasing that number. Again, in SeqMan Pro, this was hidden quite a bit. You had to go under advanced parameters and kind of go looking for it. A lot of our customers never knew it was there, and they didn't know they had control over the stringency. So we exposed the things that were we felt were really important for users to have easy access to. So now when I assemble, I get a project report, so it tells me where all the reads went. And in this Explorer panel, we have a config. So these sequences go into the config, and now I can evaluate. And you'll notice what I'm doing, there's some small icons here. Show alignment view, show strategy view, feature table. So to bring views here, I like to use these buttons because they're right. I select which config, I select alignment view. It's faster than going up to the menu and trying to select the views. So these little buttons over here are very important and save you a lot of mouse scrolling. So now we have an alignment, and we can see that there's some trace files here. I can expand them with the expandy and look at the chromatograms themselves. What's different now with 17.2 is we've got a little bit more information here with the consensus as well. So if I go, you'll see now we have two consensuses. We have trace and majority. Again, that's what we had in SeqMan Pro. Trace is a consensus that's computed from our own algorithm. It's a DNASTAR algorithm that looks at the quality of the peaks and makes a call based on the geometry of the peak, much like a human eye would. And using that trace call, it can get a consensus right, especially if there's only a one or two sequences present, a lot more frequently than just using a majority method. Here you see a case where we can show both. We called the C, majority method would have been an S in this case, where there's a C and a G. And so you can kind of compare the two different methods and see, convince yourself that the DNASTAR trace method actually gives you a better consensus than what a lot of other softwares will just use this majority. And under project parameters, I can set that consensus caller, right? So I can say, well, no, I'm going to do a majority method and switch the primary consensus now to majority, right? So there might be cases when you might want to do that. Now to search this consensus, there's a little search alignment tool, and it brings up a details bar here that allows me to do different types of searches. So one type of search is just a conflict, and these arrows will then allow me to scroll through the alignment and move from conflict to conflict. So each time I click the arrow, I get to a different place, right? Now I'm going to go back here and expand. So I right click there and I can expand all my traces. That's the fastest way to do that. Especially if you have deep data, you don't want to click these little circles many times. So I can move through and look at conflicts now. I can change this and do different types of searches. And so I can do things like a coverage search or go to a certain position. So if I want to go to the position, 150 in the consensus, I can navigate right to that position. If I want to look at a certain coverage, in some, usually not Sanger as much, but if I want to show me where I have at least 8 sequences of coverage, and it will navigate there. And again, I can, it's nice with Ultra, is I can zoom out, and get essentially a strategy view of my sequences. So if you're aligning to something that's larger, or where there's kind of sporadic sequences aligned to a larger reference, this is very useful. Or if you're looking for thin areas or overly deep areas on a de novo assembly, the coverage is a nice way to do a search. I can also search in all sequences. Now I'm going to go back to a zoom level here that allows me to see the bases. So these restore default zoom gets me to the kind of the optimal zoom. There's also a kind of a compact view if you want to see as many residues as you can. So these little toolbars, we try to put the things here that we think folks are going to use the most, right? And there's a balance between too many little tiny buttons all over and not enough. So we'll let our customers tell us when we start to have too many little buttons on our toolbars. So again, here is the alignment view. I can make changes as well to the style, but I think I'll use, so there's a whole style panel here. Assembly isn't super interesting. There's only relatively few changes. So we'll do another one that is has more differences that we can highlight. So I'm going to close this. Now, normally I would just start another project, but I know that in this build, we have a problem launching multiple. So I'm just going to try to avoid any problem right now and start a new assembly here. And I'll do a reference guided. So I went through the de novo assembly. Now I'm adding a reference sequence, and I'll talk about, I know there's a question about trimming, so we'll cover that again. So there is a reference sequence that I've added, and you can see it's checked in the reference column. And now we're going to add some reads. And now we have trimming, quality trim by default. So I can trim the sequences now, and it will give me the values. I can click on these traces and see them, right? So just like the de novo assembly, I can see where the quality trim does the trimming, and I can also do a vector trim. And this will really clean the assembly up, in terms of if you're going to try to look for some variations, you really want all the bad, low quality data and the vector trimmed. And so now I can trim by both quality and vector. And you'll see that in the unassembled table that we have these coordinates, and down in the previewer, the vector trim now is this orange color and the quality trim is yellow. And most of these sequences have vector present. Most of what's not good is vector sequence. So now I have pre-trimmed these sequences and they're ready to assemble. I also mentioned that there's a match percentage here. We expose that because it's the single most important parameter for setting the stringency for the assembly. So I'll assemble this. Oh, something is, what's going on here? Sorry about that. I think I'm losing my license file, I think. I don't know if are we still connected, Sharon? Sorry.
Yes, I can still hear you. And,
okay.
Folks that are attending, I really apologize for this. This is something that happens when we are using GoToMeeting. something about GoToMeeting can interfere with our software, or they just don't play well together. I've been using the new version of SeqMan Ultra in writing the help manual for about the past 2 or 3 months, and I have never once had it crash. So this is not something that you would experience unless you are trying to give a webinar on it using GoToMeeting.
Yeah, it's very flaky right now. Okay, so let's just go again here. Okay, so we'll add reference, some reads, and assemble. Okay, so now we have, again, we have the reference sequence with features, the trace data, and as we scroll here, we can see some areas of mismatch here. This is some vector that I left on the sequence, so you can see what that mismatch will look like. And of course, the 2 different rulers. So if you're working in reference space and you're looking for a particular location in the reference, you have the reference ruler, to find things. Whereas the consensus space will be gapped, and very quickly, if there's lots of gaps in the assembly, get out of sync. And you can see here that the numbers on the 2 different rulers are quite different. And I can search then, and go to specific reference positions. So if there's something in the reference at a certain spot, you can get right to that location very easily now. So again, the 2 different rulers really helps clarify, the coordinate system that you're working in. So that's right. Now there's another piece to the reference guided. I'm gonna go back here to close this. Go back to the welcome. So under new assembly, now what happens if I try to do something that's big, genomic in size, right? So I'm gonna add that reference. So here's a whole E. coli genome. So we'll get a message then that's gonna send this to Engine. And so it's just detecting that this is a larger project. You'd be better served, going, through the wizard, right? And now it automatically loads that reference in, and I can now set up an assembly here. So it's more clicks, right? It's gonna ask you for more information, but it also gives you feedback to make sure that, your particular computer is gonna be adequate for what you're asking it to do. So, and that's stuff that we've covered in other other webinars here. So this just goes through that full wizard. I expect that's a relatively few Sanger users are gonna do this. It's mostly gonna be for NGS or for the relatively few that are gonna align to whole, chromosomes, for instance. So it's a kind of a safeguard then for folks that have very large projects to kind of catch that and make a redirection for you. So I'm gonna show you one other type of project here, and that is using this checkbox, allow adding single sequence contigs. And this is a parameter that was really hidden in SeqMan Pro. And what it allows us to do is run an assembly where what our goal is to try to cluster things that are similar, but make separate contigs out of them, rather than lumping them all together. So I'm gonna add some sequences that are derived from antibodies, from antibody sequencing. And of course, they're from the same region, so they're quite similar to each other. I'm just gonna load a set of these. But they have variable areas, roughly in the middle. And so if I assemble at 80%, they'll just mostly just lump together, right? And that may be okay for some projects, but I might also want to try to parse them. And I can do that by increasing this stringency. So I'm going to increase it to 97. And I'm also going to say, allow adding single sequence contigs. It's possible that some of these sequences don't really match any of the other ones very well, but they're still interesting to me. They're just another antibody that's a little bit different. And so this parameter then, when I assemble, will show up. Now you can see this takes a little longer because it's doing a lot of parsing here and trying to fit these. And you can see just from the project report, I'm just kind of scrolling through here. You know, it's working pretty hard trying to place these ABI reads into different clusters. And as I expand, you can see now that we've got 26 contigs, and some of them have up to 3 sequences in them, and some only have a single sequence. And for example, in this workflow, I might export the consensus out from these. It may be the consensus of these that I'm interested in. I'm just using the algorithm then to group them together. Now, when I look at the assembly, you know, I can see areas that are even at 97% that are still different, right? So I might decide to go back and redo this at 99% if I really want to parse these out and try to get almost no mismatch between the sequences in the contig. So it just gives me that flexibility. Now, when I have different, let's try this a little. Yeah, I guess I can use this one. So we can also go to our style now, and there's more control in Ultra over how we want this to look. And so this is kind of a basic default that we use, where we just highlight differences as yellow, but there's other things that we might do. And, you know, for example, we might show only differences from the consensus, especially where the sequences are mostly similar. Sometimes this is a better way. And you can do things like, you know, color the background, you know, and use some of these different, that you often see in multi-sequence alignments. But we brought this over to SeqMan 2. So depending on the type of image you want, you know, you might want to use SeqMan to really highlight it in a certain way. So we brought some more functionality into SeqMan Ultra that allows us to highlight this. Now, if I want to take an image of this and export it, we have a little camera right here that exports the image. And so I can save this image now. Here, I'll just name it something else. Let's see, antibody 97. So I can save it as a PDF, as a PowerPoint, or as 2 different types of bitmaps. And so PDF is all right, but I'm gonna go and do a PowerPoint, which is kind of unique. So we're gonna go and it'll ask me, it's essentially a WYSIWYG. So what you see is what you get, but I can set a different range of views, you know, to try to alter what's actually exported. And now I'm just exporting this to a PowerPoint, and let's go look at, and the reason I like PowerPoint, of course, it's good for embedding in PowerPoint presentations, but it gives me a little bit more ability to edit the image. So let's see, it's this one here. So we open this in PowerPoint, and it looks pretty crisp. And what this is grouped objects now. So I can go in here and ungroup. And now if you're trying to get this, you know, if you wanted to use this image, you know, there might be some aspects here, like this line, that you don't want. And so you can go in and select those little pieces there that you might not want in an image that you're trying to publish. And here's like another little line up here. So it's kind of a low-tech way to alter your image. And of course, you could, you know, with the PDF, you could go to more advanced graphics programs and make edits. But PowerPoint, for the basic user, it's really nice. I can just go in here and of course, add additional annotation using PowerPoint very simply. So really a nice functionality. It's in Ultra, it's in SeqMan Pro, it's in GenVision Pro, something that you'll see us continue to do. Okay, so that's exporting image. I can also add more data. So, you know, if I have more data that I wanted to add to this assembly, you know, this is a pretty big data set. You know, more reads. Right, so it gives us that unassembled window, gives us the ability then to add more data. I can also, I didn't show editing. I know that was shown in some earlier webinars, but there's editing capability as well. So when we go back to our sequence view, let me just go back to style and just show all editing is allowed. And let's see, so I can go in, I'm just going to place a cursor so I can go in and backspace, edit, I can type, so you can select areas. I can select, if I select on the consensus, that will select a whole stack of sequences if I have a stack, but I can delete, chunks of sequences. You can also use it on the edges. Let's get in, what's more common, I think, is, if we have a config that's, I can select and trim up a ragged edge, potentially. So lots of different edits that we can do here. And then there's bigger edits as well that we don't do with Sanger so much, but at genomic scales, we can split contigs, merge contigs, that sort of thing. Okay, well, now we're at a little past a half hour, and I think that's about all we intended to cover today. I'd be more than happy to take any questions. Hopefully we'll keep our connection going here. So if I don't get to your question, Sharon will have them, and we'll certainly respond, via email as well.
Hi, Matt. I have some questions for you. I was actually just typing an answer to one of our viewers, Elena, and maybe you can just answer this in person. Sure. So Elena asked if it's possible to change the order of sequences in the assembly view window. In SeqMan Pro, you used to be able to drag and drop, but you can't do that. So I was just starting to explain about the sequence sort reads command.
Oh yeah, so there is, so we can sort the reads, but we can't manually move them around yet. That is, I think we've gotten that feedback, and it's in, on the development board, but I don't know off the top of my head if it's, I don't think it's in this next update, but it's definitely made it to our development board.
Okay, but you can sort by
here is a
name.
Little, yeah, here is, so this little toolbar, I can sort by name, and then I can sort by position, which is what the default is. But I don't, well, let's try. I don't think, yeah, so I'm trying to grab and drag. We can't manually move these order of the sequences. But I think we'll be able to get that. That seems like something that we can do in other areas as well.
Yeah, so that's something that we can do in our kind of similar interface applications. So I don't think that would be a big deal to be able to put that in. But anyway, we do have 3 different ways to sort automatically, but you can't do it manually yet. Okay, so Paul asked a question about using SeqMan Ultra to analyze PacBio data, and I replied that you need to do use SeqMan NGen because that's an NGS assembly. But then the second part of his question, I'm not sure if this applies, but I thought I'd ask it anyway. If you analyze PacBio data using SeqMan Ultra, so it's analyzing an NGS assembly, can you use it to detect methylated nucleotides?
Right now, the answer is no. So there's two parts to the answer. One is to work with PacBio data. That's considered, a genomic workflow. So if I do, and right now in today's build, we have options for de novo assembly of PacBio. Our current development team is working on templated, reference-guided PacBio assembly. And so that's what we're working on now. We expect to have that early in 2021 to have, for SNP analysis, finding variants, structural variants with PacBio data. In particular, the PacBio data, the newer data, the CCS data and the HiFi data, essentially the quality is like Illumina. It's long, they're long reads, 10,000 base pairs or more, really high quality, and you can do very accurate variant analysis with that PacBio data. Now I forgot the second part of the question. So we can do the alignment there. Oh, the methylation.
Yeah,
methylation. I know that it's different than the bisulfite sequencing that we did with Illumina, and it's in the development board, but I don't think that would make our first round. But that's hard to say. If the bisulfite is detected right as the read is being constructed, then it's possible we could get it sooner. But yeah, it's not going to be in this first version of our PacBio templated support.
Okay. Next question is, if I go into an ABI file, just open up the file in a, I don't know, a text editor or something, and delete some of the poor sequence from it.
Yeah.
And then I try to open that in SeqMan Ultra, I get an error message. So do I have to use an unmodified ABI file?
Yeah.
And do the trimming?
Yeah, they're a little tricky, the ABIs, you know, so ABI format, I don't believe supports trim points at all, or AB1 format. I think what happens, you have to convert them to SCF format that has some trim information inside. But generally, when we do trimming on ABI, we store that information outside the file in a little auxiliary file that says, here's where all the trim points are. So I don't think that, I have not seen ABI files with trim points built in them that we can read, at least out of user sets. So unless there's some very new format that allows trim points to be set inside the file.
Okay.
I don't know if that was much of an answer, but it's confusing for everybody. I know that.
So let's see here. Leonel has a question. Can you compare the quality scores for each of the runs when obtaining contigs when you increase the minimum match value? I'm not sure I get that question. We might have to do that by email.
Yeah, I'm not quite sure.
Leonel, we'll get back to you by email with more information about that. Okay, Paul has a question about exporting images of long alignments, like hundreds or thousands of base pairs. Can you export images of something that long?
There's a limit, right? It's because it's, you know, if it's bitmap, you know, it gets quite large pretty quickly because it is an image. So there's a limit. I don't know exactly what that limit is. Seems like I've hit it with PDF. I've tried it with big things with PDF, and they hit a limit at a certain point. So, you know, it's hundreds, yeah, thousands or hundreds of thousands, probably not. Is what I would guess.
Okay.
At least at the Zoom, you know, if it's hundreds of pages, it's probably going to run out of juice.
Diane has a related question, which is, what's the best option for exporting an alignment that's about, you know, a kilobyte in length or an entire gene? Can you export it to Word?
So exporting the actual aligned data with the reads and everything.
Yeah.
You know, those are kind of tricky. One thing would be there's two formats that I can think of. One is not supported anymore. It's a ACE format. So you used to be able to save, you know, an assembly into ACE, ACE, and some different programs could open it. That's pretty much gone away. We don't support it, and we haven't seen any for a number of years. So the other format is exporting as a gapped FASTA. But that's usually just the consensus, if it's gapped or not. So there's not really, you know, there really isn't a good option for exporting the actual alignment into some other format, just because they contain so much information.
Mm-hmm.
Each read, the name of the read, the gaps for the reads. So there really isn't, you know, there's BAM. So maybe BAM is, but you can't really look at BAM. It's a binary file. So
yeah, it's not human readable.
No.
Okay, well, that was the last question that we have.